Okay, so it's my pleasure to introduce Hank Baker, Dr. Hank Baker, seminar speaker today. But before doing that, I actually want to introduce you to Caitlin Williams, who is a new PhD student in Coastal Oceanography Group and in Hydrologic Sciences Graduate Group. And in particular, she did all the work to coordinate the seminar and set up Hank and so on. So she's the one to thank. Our guest, Hank Baker, is an evolutionary ecologist with an affinity for freshwater and anadromous fish. That's very cool. He's presently a postdoc at Berkeley in the Environmental Science Policy and Management Department. Got his undergrad degrees, as I told you I was going to do these universities. Brown University, he's got one stamp from them, and his PhD from UC San Diego. And I think you're going to expand a bit more on who you are, but key point is that your research seeks to understand the drivers and intraspecific trait variation and its implications for population, community, and food web dynamics, as well as applications to conservation. Focused on imperiled fishes, working in Coho, now Central California Coho to be more specific, and even more specific in the context of the Russian River. Thanks for visiting us, sharing your research. Nice to have you here, and yeah, please feel free to provide some more background to who you are before you dive into the real geekies. Okay?
All right.
It's over to you.
All right, thanks so much, John.
Sure. Dim the lights. People can see you. Well, maybe we'll dim them in a little bit.
How's the sound? Can everyone hear me okay? Cool. All right, well, yeah, just thanks so much to John for inviting me up here, Caitlin for showing me around, all the folks that have given me tours and met with me and talked to me about their research today. Just really delighted to be here. So as John mentioned, my name's Hank. I'm going to be talking about some of my recent work on variation in fish movement strategies. John gave a little bit of an introduction to my background, but I'll just give a little bit of personal introduction here. I grew up in Denver, Colorado, and learned to fly fish with my dad and became just totally obsessed with catching fish as a kid. And I think this is sort of how I got into fish ecology, but it took a long time to sort of materialize. You know, as I learned to catch them more, I became interested in the places that they live, their individual behavior. And then fast forward to undergrad, I went out to the East Coast at Brown University and didn't immediately become an ecologist. Spent my first couple years studying neuroscience and human cognition, and then took an ecology class my junior year, and it sort of dawned on me for the first time that people can make a career studying fish. And I was like, why don't I do that? That's awesome. So I was very fortunate to get an internship at the Marine Biological Lab studying striped bass in salt marshes, and that sort of launched my career into fish ecology. And then after graduating, I got to spend a summer out in the Arctic on the north slope of the Brooks Range at Toolik Field Station as a limnology tech for the long-term ecological research site. Got to get my hands on some fish while I was up there and grow a nasty looking beard while I was up there as well. And then moved to the Bay Area after that and started working for a company called Stillwater Sciences. Spent about 3 years there doing fish ecology and limnology related projects, and then decided it was time to go get my PhD. So that brought me down to the University of California, San Diego, where I worked with John Shuren, who's a community ecologist and food web ecologist who does a bunch of work out in the Eastern Sierras and the lakes of Yosemite National Park. So that was an easy sell for me to live by the ocean in Southern California and do my field work in the mountains. So I did most of my research at the Sierra Nevada Aquatic Research Lab, which is near Mammoth Lakes, California. Mostly studying fish. It's in particular a unique little fish called the tui chub, which is a native minnow that's distributed all throughout the Western Great Basin, and also got to study some trout introduced to high mountain lakes in Yosemite National Park. And then in 2023, I finished my PhD, which brought me to UC Berkeley, where I am delighted to be a postdoc working with Stephanie Carlson and Ted Grantham, and doing work just up the road in the Russian River watershed. And I've really loved getting to know this area and this little part of the world. And thanks for sharing it again with me today. Okay, so just a quick outline for my talk. Now that the personal introduction's done, I'll first kind of give an introduction to something that I'm going to talk a lot about today, which is intraspecific variation. What is it? Why does it matter? Why am I so interested in it? Then give a quick overview of sort of my research interests and the approach that I take. I'm not going to have time to go into depth into all the things that I'm interested in today, so I kind of want to give a bird's eye view of my broader research agenda, and then talk in depth about a few projects, including some of my current work, 2 projects from my Russian River work, and then one back from my PhD to kind of tie it together. And then hopefully, if I have time, a few minutes for some of the work that I'm interested in doing in the near future. Okay, so what is intraspecific variation? You may recognize this fellow here, Charles Darwin, said that they are the many slight differences being observed in individuals of the same species inhabiting the same confined locality. You may be familiar with this. A familiar example might be the domestic dog, which exhibits fairly extraordinary variation in morphology and behavior as a result of selective breeding by humans. You may also be familiar with sexual dimorphisms like these mallard ducks, or ecotypic variation like these benthic and limnetic sticklebacks. So these fish over here on the right are roughly the same age and come from the same lake. The fellow on the top lives on the bottom and eats big bugs, and the one on the bottom lives on the top and eats zooplankton. Intraspecific variation comes in very many forms, is the point of this slide, and that can include continuous traits, discrete traits, and one key element of variation is how animals move, which I'll be talking about today as well. Okay, so now why does intraspecific variation matter? Why do we care about all this? Darwin says these individual differences are of the highest importance to us, for they are inherited and thus afford the materials for natural selection to act on and accumulate. We've since learned that that intraspecific variation is not only the substrate for selection, but can be the outcome of it as well. Oh, that one came out weird. Darwin went on to lament that most naturalists of his time often considered these individual differences relatively unimportant, and it took ecologists a very long time to realize that they are in fact important, and this work has been sort of accelerating over the last few decades. Some papers that sort of piqued my interest in this were Dan Bolnick's paper from 2011 highlighting some of the ways that intraspecific variation can affect community ecology. Here's a somewhat recent meta-analysis by DeRoche and colleagues. And on the x-axis here, we have species effects, and on the y-axis, we have intraspecific effects. And what I want you to see is that many of these points are at or above this 1-to-1 line, so indicating that intraspecific variation can have ecological effects that can equal or exceed those of species changes in communities. And today I'll be talking a lot about movement and a lot about movement in fish, but I want to point out that intrapopulation variation in movement is common across basically everything that moves. So if you study something other than coho salmon or the fish, I hope you can find some of what I'm talking about relatable today. Okay, this one's coming out a little weird too, but I generally consider myself an evolutionary ecologist. I'm interested in sort of the interplay between ecology and evolution. And within that broad conceptual framework, I'm particularly interested in how ecological interactions and environmental conditions shape trait variation in populations, and in turn, how trait variation can affect the dynamics of populations, communities, and food webs through mechanisms like portfolio effects, Jensen's inequality, or changes to the strength, number, or identity of trophic interactions. And so like I said, I don't have time to cover everything that I'm interested in, but some of the approaches that I've used in my past work have been comparative surveys of diet or resource use using gut content analysis or stable isotope analysis surveys, behavioral assays with wild-caught fish brought into the laboratory and then characterized for their behavioral phenotypes, morphometrics, genomics, mark recapture to enumerate and follow the movement of individuals, and then manipulative experiments in mesocosms or in situ enclosures through transplants. So I'm just going to be highlighting a few of these today, but I'd be happy to talk more about any of this stuff if you'd like. Okay, a few key takeaways that I want you to get from what I'm about to tell you in this talk. First, that juvenile coho salmon are much more diverse than is typically appreciated. And that even subtle diversity, and maybe subtle is not the right word, but fine scale diversity, can have large effects on population dynamics. Third, that phenotypic variation, including in movement and rearing strategies, interacts with key dimensions of landscape heterogeneity or variation in environmental conditions across the landscape. And finally, that managing for this diversity can improve restoration and conservation outcomes. And finally, the sort of other side of this coin is that behavioral variation can also have effects on other things that we as ecologists consider important. So I'll show through a mesocosm experiment that just changing the behavioral composition of fish can have pretty striking effects on the spatial structure of food webs. Okay, so I'm going to be talking a lot about salmon today, and many of you may be familiar with this, but in particular, I'll be talking about the endangered Central California coast coho salmon. Coho salmon range from Central California, just south of Santa Cruz, around the Pacific Rim to Hokkaido, Japan. They are evolutionarily distinct at different points along their range, including in Central California, where they're protected as an endangered species. The Central California coast, ESU, includes the Russian River Basin right here, which is the largest watershed within the Central California coast and historically supported the largest populations of coho salmon. Populations have since declined drastically following colonization, European colonization, and subsequent land use change. And beginning in the late 1900s, populations approached zero. So now there's, over the last several decades, there's been some pretty empowering restoration and conservation efforts to bring fish back into the Russian River Basin. That included the founding of a conservation hatchery program, which took local broodstock, raised juvenile fish, and has been releasing juvenile fish into some 26 different tributaries of the Russian River Basin over the last couple of decades to try to sort of rekindle these wild reproducing populations. Bless you, John. And along with that, there's been a lot of effort to monitor how these fish are doing. And as part of that, we have been tracking fish through pit tag antenna monitoring equipment, which I'll go into more detail later. But these red dots here represent some of the sites where fish have been monitored over this time. Okay, so many of you may be familiar with this diagram, but I'll give sort of an overview of the typical coho salmon life cycle. So typically, age 3 adults return to their natal stream, the stream where they were born, to spawn in the late fall and winter. Eggs incubate, and then free-swimming fish hatch out in the spring. And then these fish typically rear to where they're born for a period of 1 year before smolting, swimming out to sea to complete their life cycle. However, this is sort of the modal pattern or the most common pattern of their life cycle. But we've since learned that there's variation at virtually every stage of this life cycle. My colleague Gabe Rossi likes to call it more of a tangled knot than a circle diagram. And that includes variation in their juvenile life cycle, which they complete in freshwater streams. And one tool, which I briefly mentioned earlier, that's been really useful for understanding this variation in their juvenile life stages has been pit tag detection systems. So for those of you that have never seen this before, these are tiny RFID chips, like what you might have in your fast track on your car or something like a ski pass. And you surgically implant these into the belly of the fish, and then you set up these monitoring arrays at areas that you're interested in. And so those are shown by our red dots in the Russian River Basin. And then every time that fish passes the antenna, it pings an individual identifier and a timestamp, so you can sort of track their movement at key points in space and time. Okay, so now with that background, I'm going to be jumping into some of my research, which is going to start by talking about a lot of this pit tag detection data. And I have sort of 3 key parts to this portion of the presentation. In part 1, I'll be talking about how variation in movement within a single creek can affect population dynamics of coho salmon. In part 2, I'll kind of zoom out across the watershed to try to understand how variation in habitat conditions across the riverscape can affect the frequency and phenology of what is called non-natal rearing in juvenile salmon. And then finally, for part 3, I'll be talking about how variation in behavioral strategies can then in turn affect food web dynamics using a mesocosm experiment that I did. Okay, so first I kind of want to introduce this idea of portfolio effects. So for those of you fortunate enough to have financial portfolios during these trying times, you might know that having a well-diversified financial portfolio stabilizes the portfolio yields through time. And the same is true in biological systems. So this is an example of metapopulation dynamics by Dan Schindler and colleagues, showing that the metapopulation of sockeye salmon, shown by this thick black line here, is stabilized as a result of diversity of its constituent populations, shown by these little squiggly lines here. And you can see that this black line is flatter than sort of the squiggly dynamics of its constituent parts. So these ideas kind of make sense. I think that diversity can confer stability, but what we're interested in understanding is, like, do these stabilizing effects emerge at much smaller scales of biological variation? And to do that, we looked at variation in rearing strategies within a single creek of coho salmon. So again, I've shown this map a couple of times now. This is the Russian River watershed. We're going to be zooming into this bottom one here, which is called Willow Creek. This is the lowermost major river left tributary to the Russian River. It is a really cool place. So if you all are up here, this is just down the road. And it's a really beautiful place. There's an awesome hike from Shell Beach over to the Pomo Creek campground that I encourage you to check out. And it is teeming with salmon this year. Okay, but that is an aside from the talk. So Willow Creek is a really interesting place, in part because there's these really stark habitat transitions over its length. So if you go into the upper reaches, up where the historic spawning habitat was in Willow Creek, it looks like what maybe you think of when you think of a coho stream. It's flowing through second growth redwood forest. It's cold. It's got large woody debris thanks to some good restoration efforts in the creek. And it's sort of a single meandering channel with spawning gravels. If you follow that about halfway down the creek near where one of our monitoring sites is, the river changes pretty dramatic, or the creek changes pretty dramatically. It spreads out into a broad and flat alluvial valley that has some really dynamic and braided channels. It's much slower flowing through this area. There's some kind of complicated and changing channel shapes, and a very thick alder forest. It's pretty cool to be inside. But all in all, we think this might be really awesome habitat for coho salmon. There's tons of submerged terrestrial vegetation, which you can see in this when the creek flows come up. We think that there's a lot of bugs, terrestrial bugs that come in, and a lot of high productivity. And then below that, it flows into this tidal wetland, which when the tide comes up or when there's backwater flooding, when the Russian River mouth is closed, becomes inundated, again, might provide some unique foraging habitat for juvenile coho salmon. Okay, so again, we're looking at pit tag detection data here, and we're gonna be looking at detections at two different antennae. So the first one sits kind of right at this habitat transition between the what we're going to be calling the natal habitat, or the spawning habitat in this stream, and then below which sits this kind of unique different habitat in the lower portion. And so when individuals pass this first antenna, I'll be referring to that as down migration, and we'll be looking at the timing of down migration. And then when individuals pass out of Willow Creek altogether and swim into the Willow Creek estuary, we're going to be referring to that as emigration. Okay, so I'll kind of populate this research question slides with questions and answers as we go through this. The first question we had is, do juvenile coho salmon within Willow Creek have variation in their movement, phenology, and rearing strategies? So to do that, we looked at the timing of down migration and find this really distinct multimodal pattern here. So on the x-axis here, you have the water year day, or days since October 1st, that the fish passed from this habitat, where they were either born or placed by the conservation hatchery that contains all the spawning habitat, when they pass down into this portion. Okay, so you can see one large peak where they do sort of what I referred to as the typical coho salmon life history pattern, where they stay near to where they were born or placed by the hatchery until the springtime, when it's time to swim out to sea. But a subset of these fish, and you can see two modes here. If you break it up by year, it's actually just one sort of fall mode, but these fish leave their natal habitat early and down migrate in the lower portions of Willow Creek to spend an extended residence time beginning in the late fall and winter. And so we'll be calling those fish non-natal rearers for this particular study. Okay, so do they exhibit variation in movement? Yes, very clearly, distinct bimodal pattern in their timing of movement. Okay, so next we wanted to understand is, are there consequences of this for population dynamics? Here we have the proportion. Yellow is going to be our natal fish. Blue is going to be our non-natal fish, or the fish that moved down early. And then this is just their composition as juveniles. You can see that it is marginally less common, so it's about 40% of the population exhibits this non-natal rearing strategy. But when we look at adult returns, we find that they are overrepresented among these adult returns, indicating that this non-natal rearing strategy is adaptive in Willow Creek. Okay, so next we're interested in trying to understand whether it affects population stability. And here I'll show you a plot with a measure of stability of population of adult returns over time on the y-axis, and then we'll have the natal rears alone, the non-natal rears, and then the whole population here. And what we can see is that the natal rears had the most variable populations over time, or the least stable, and that the non-natal rears were most stable through time, which had a net stabilizing effect on the population as a whole. And this is a pretty strong effect, 1.4x. So what are the consequences of variation for population dynamics? You get more adult returns. So non-natal rears come back at a higher frequency than their natal rearing counterparts, and that has a stabilizing effect on population dynamics. Okay, so why are these non-natal rears coming back more often than the natal rears? We think part of the answer might lie in their timing of emigration and their growth rates, and so I'll show you those now. So on the y-axis, we have the water year date at which they emigrated or left Willow Creek for the Russian River estuary. And what you can see is that the non-natal rears left considerably earlier than their natal rearing counterparts, but also had much more variable patterns or much more variation in their timing of emigration. And so we think this might be important because the timing of emigration is a critical life stage for juvenile salmon. They're transitioning from life in fresh water to life in the sea. They're vulnerable to predation, and they need to grow quickly. And so timing up their ocean entry with, in particular, conditions of coastal upwelling lead to rapid growth and can have pretty profound effects on their subsequent survival. And so we think that this might be sort of another level of a portfolio effect here, where just having a broader distribution might allow them to have more individuals that sync up with those good conditions in the coastal ocean for rapid growth. Okay, and then we only have 1 year of growth data on these fish, but what you can see from this is, so this is daily growth on the y-axis here, and what you can see is that the median growth rate between these two strategies was almost identical. But again, we see much more variation in growth between in the non-natal rearing conditions. So you get higher maximum, but also lower minimum growth. And perhaps some of these individuals are what's represented in the adult returns. Okay, so variation in emigration timing and variation in growth provide sort of plausible mechanisms for why non-natal rears come back at a higher frequency than natal rears. And then one final question, we're interested in trying to understand how the number of non-natal rears varies over time and whether that's related to flow within the river. Okay, so on the y-axis here, you see the proportion of fish that exhibit this non-natal rearing strategy. Each point represents a year. And what you can see, and then on the x-axis, we have flow. So this fit is, it's an okay fit, but you don't need statistics to see that in years where there's very little flow, you get almost no fish that exhibit this non-natal rearing strategy. Okay, I don't know why this is looking like this. So is there a relationship between movement variation and flow? Yes, variation in rearing is lost during drought years. So in addition to all the other bad things that come with drought for fish, we think that one sort of additional effect is that population instability might be exacerbated by the loss of this critical phenotypic variation. So something for us to keep an eye on. And if you want to learn more, I encourage you to check out our recent paper in Ecology Letters on this topic. We're doing on time. Okay. Okay, so that concludes part one. In part two, I'm going to zoom out from just Willow Creek to look at how variation across the riverscape affects the frequency and phenology of non-natal rearing. Okay, so this first study, I was looking just here at movement in their creeks, but fish not only move down and stay within lower portions of their creek, but some individuals leave their natal creek altogether and find different areas of the river network to complete their juvenile life cycle. We were interested in how watershed characteristics, sort of characterizing these patterns. How do watershed characteristics affect where these fish go that leave their natal streams? And we were fortunate that that the sort of long-term monitoring sites that have been set up over the last sort of 14 years or so set up a sort of nice experimental gradient for us. So we have 4 life cycle monitoring streams that have been intensively monitored over the last 14 years. And they are Willow Creek, which is the lowermost tributary here, which is a low gradient stream, so it's relatively flat across the course of its stream. And then the next is Dutch Bill, which is a high gradient stream, so it's a little steeper, which is a pretty good correlate for various habitat conditions. And then Green Valley Creek, which is farther away from the estuary, and another low gradient stream, which I'll be using green to show. And then the farthest away is Mill Creek, which is a high gradient stream as well. So we get position within the river network. So this is nearest to the estuary, farthest from the estuary, and then sort of alternating high and low gradient streams. Okay, and so to do this study, we took the entire database of fish that had been released of pit-tagged fish that had been released into Willow Creek, which represents about 15% of the total releases. This included 280,000 individuals with nearly 2 million observations. And then we filtered for fish that were detected in one of these 4 focal streams that were released or tagged in a different stream. This yielded about 31,000 observations of about 2,000 fish. And one sort of just a side point here is that this is a rare trait, but as we learned from the last section, variation is really important for population dynamics. And so it takes a ton of data to try to understand these sort of rare traits that might be really important for population dynamics. So okay, so the first question was, do stream gradient and position of these streams within the river network affect the number of fish that use them for non-natal rearing? And I'll show 2 plots here. The first is going to be lower antennas. So these are the antennas within each stream that are near to the confluence. So this shows when a fish enters. And then upper antennas that show when a fish has migrated several kilometers up into the spawning habitat of those creeks. Okay, so what we can see from here is that Willow Creek is used about 5 times more often than any of these other creeks for non-natal rearing. This is effort standardized over here, so don't pay too much attention to the particular units. And you can see that, well, now pay attention to the units. So this is much less common than rearing in the lower portion. So very few of the fish that enter these non-natal streams actually make it all the way up into the upper portion of the rivers. But of those that do, they really only do it in these low gradient streams. And so this is about 10 times more fish than in these high gradient streams, which makes sense. High gradient streams are hard to swim up. Fish are probably going to be using low gradient streams to swim far up them. Okay, so the first research question, do stream gradient and position within the river network affect the frequency of non-natal rearing? Yes, low gradient streams nearest to the estuary attract 5 times as many non-natal rearers as other sites. And does it affect the location of non-natal rearing? Yes, fish really only reach the upper tributaries in these low gradient streams. Okay, so next we're going to be looking at their timing of use of these non-natal streams. And in particular, when they exit and when they enter these streams and when they exit them. Okay, so this plot shows across all our streams our timing of entry of these non-natal streams, and what you can see is that there are sort of 3 distinct modes that correspond to 3 distinct juvenile life stages. So these individuals here are in their and this is, I should mention, these are days since March 1st, which is approximately days since they hatched or their age. So these fish here are entering in their first spring. These are what we call fry. These fish are entering in their first fall or winter. These are what we call par. And then these individuals are entering their second spring around the time that they're preparing to swim out to sea. And these are what we call pre-smolts or smolts. Okay, now we're looking at the timing of entry, shown on the left here, and the timing of exit from their non-natal streams on the right. Again, the purple is going to be our low gradients or our high gradient streams. The green is going to be our low gradient streams. And what you can see from this plot here, maybe I'll draw your attention to the box plots here, is that the timing of entering the non-natal streams is later in low gradient streams, and the timing of exit is also later. So these fish are arriving at and leaving from low gradient streams later than high gradient streams. So next we were kind of curious, like, is this effect happening within life stages, or is it changes in the composition of life stages that these streams are attracting? We can see a pretty small but significant effect of within life stage delays in the timing of entry to non-natal streams in low gradient streams. But what we really think is driving this effect is differences in life stage composition. So again, this is Willow Creek and Green Valley here are two low gradient streams. These correspond to the green plots over here. And what we can see is that they're attracting these later life stages more often. Okay, so do stream gradient position affect the phenology of non-natal rearing? Yes. Low gradient streams have earlier arrival and departure. And this effect is mostly driven by attracting proportionally more spring migrants, especially pre-smolts and smolts. Okay, and then sort of a bonus question. We didn't really have any a priori hypotheses about this, but we looked at the time of day at which they entered these streams. And for juvenile fish that are susceptible to visually foraging predators, you might expect that they would move more often at night. And the literature kind of indicates that this is true. And in fact, when you plot this, you see some hint at that effect. And then this is a QQ plot showing the sample quantiles relative to a theoretical uniform distribution. And what you can see from this is that between about 7 a.m. and 5 p.m., you see less movement than would be expected under a uniform distribution. But when we break that up by stream gradient, we see that this is really only true in high gradient streams. So in Green Valley and Willow Creek, they're entering streams pretty much uniformly across the course of the day, whereas in these high gradient streams, fish are moving into them selectively more often at night. Don't know what's going on there. I'd love to hear your ideas. Okay, so that concludes part 2 of this presentation, and now we'll move to part 3, where I'll be talking about sort of the flip side of this coin, how variation, phenotypic variation within fish, and in particular their behavioral strategies, affects food web dynamics. Do a quick time check. All right. And for this, we will be talking about this sort of food web here. And I'm going to move you away from coastal rivers, away from salmon, 300 miles southeast over to the eastern slope of the Sierra Nevadas, where this humble little fellow here, the tui chub, lives. I think I mentioned that this is a native minnow that occupies a bunch of inland drainages throughout the Western Great Basin. I've done some work on tui chub and sort of what makes them cool and interesting. For this study, they are just a tool for us to answer our basic question. And in particular, they have repeatable behavioral traits that we can manipulate in experimental settings. So we set up these sort of food webs in pond mesocosms, which are just 1,000-liter cattle tanks that we fill with water. And then we manipulated the behavioral composition of tui chub in these tanks. So in addition to tui chub, we have a hungry trout, adult trout predator that can eat these guys. These juveniles are about this big. They're small and totally palatable to a large adult trout. And tui chub are eating either zooplankton or macroinvertebrates. And we have sort of two sides to the food web here. So zooplankton eat phytoplankton, and these benthic macroinvertebrates eat attached algae or periphyton. And so what we're interested in here is if we vary the behavioral composition of tui chub, can we see effects on food web structure? And so I'm going to be using the term trophic cascade here. And for those of you not familiar, a trophic cascade is where changes at one trophic level, so the tui chub here, affects changes at a non-adjacent trophic level. So this would be the tui chub eats zooplankton, which releases phytoplankton from abundance. So the trophic cascade would make lakes more green because there's more phytoplankton. Does that make sense? Okay. Okay, so now a little bit of our experimental design here. We first had to characterize the behavioral traits of these individual tui chub. And to do this, we set up a somewhat silly but repeatable and effective behavioral assay to test for their boldness. And to do this, we put the fish in an aquarium with a small refuge in it. We let them get comfortable, and then the experimenter feigns a predator attack by jabbing an aquarium net towards them. The fish uniformly take cover in this half piece of PVC pipe that we have set out for them, and then we record how long it takes for them to emerge from that tank. Bold fish come out very quickly after being scared, and shy fish take much longer to come out. So we classified fish as either bold or shy based on the results of this behavioral assay, and then we manipulated the composition of bold and shyness in these pond mesocosms. We also set up some spatial structure in these mesocosms so that the trout didn't just eat them all in this tiny little bucket, and in part because it's part of our experimental design. So this little thing here represents, we're calling it a semi-permeable partition, but it's just a piece of mesh that the tui chub can swim freely through, but that's too fine for the trout to access. So that results in a refuge habitat over on the left side and a larger but risky habitat over on the right. And we had 3 treatments. It was either all shy fish, shown in red, all bold fish, shown in blue, or half shy and half bold. And then we had a couple of controls where we only had a trout or no fish at all. And then to understand the effects of this on food web structure, we measured chlorophyll A, which is a measure of phytoplankton biomass, so that's here. Periphyton biomass, so that's these guys here. And then invertebrate abundance, composition, and biomass as well, which I am not going to go into. And then we also measured, of course, tui chub growth rate and mortality inside the tanks as well. You can imagine a couple of different scenarios. The first is that bolder individuals would exert stronger top-down control or have stronger trophic cascades than shy individuals because they're more willing to go out and forage despite this, the presence of this predator. So they would eat more bugs, which would mean that less algae is being eaten and it gets greener. Alternatively, you might imagine that bold and shy fish sort of have complementarity in their feeding. So maybe the bold fish go out and feed any time of day, and then the shy fish only go out at night, or maybe the shy fish forage in the refuge habitat and the bold fish forage in the open habitat, in which case you might imagine more complete grazing of these zooplankton and invertebrates and stronger top-down control by the mixed treatment. Or finally, you might imagine that bold and shy fish exist along this growth mortality trade-off where bold fish grow faster, but at the risk of being eaten more. And so, and shy fish, they hunker down, they don't go out and forage, they grow less, but they survive more often. In which case, it's sort of unclear what we might expect in terms of the strength of the trophic dynamics. Okay, so first I'll be testing, this hypothesis here. Do we see evidence of a growth mortality trade-off in Tui chub? Again, bold are going to be shown in blue, red are going to be the shy individuals, and then we'll have our treatments down here. Your eyes might tell you that that there's a hint of an effect here where the mixed treatment fish grew faster. That was not supported statistically in our analysis, so it may just be an underpowered effect that's going on there. And again, you see a hint of an effect where bold fish might be dying more often, but again, this is not statistically significant. So we found no evidence for a growth mortality trade-off, but we're seeing hints of an effect, so maybe this should be checked out in further studies. So is there evidence for a growth mortality trade-off? No, maybe underpowered. Okay, next, does fish behavioral composition affect the strength of top-down control in these food webs? And again, top-down control is going to be measured by the response of these guys down here to changes in Tui chub behavioral composition. And first, we'll look at the right side of this food web here. So these are phytoplankton. And we can see, indeed, that bolder fish had stronger releasing effects or stronger trophic cascades on phytoplankton, with mixed treatments having the lowest effect. And for periphyton, we see no significant differences across our treatments. Does behavioral composition affect the strength of top-down control? Yes, bold fish produced stronger pelagic trophic cascades. But again, this was measured at aggregated across both the risky and refuge habitats. So next we wanted to understand if there was differences in food web structure between the risky and the refuge habitats. And so here we'll be looking at the differences between the purple and yellow for each treatment. And you can see there's slightly more top-down control in the refuge habitat. Individuals are foraging more often or more completely in that habitat because it's safer, presumably. But those effects are not significant. But for periphyton, we see very strong spatially structured effects where individuals forage much more completely in this refuge habitat, and that effect was much, much stronger in the shy fish treatment. And the reason that we think that is because shy fish are, again, foraging more often only in the risky habitat and more completely grazing down the consumers in that area. And just to give you a sense for what this looked like in real life, so this is the refuge habitat here. This is the risky habitat. So you can see that there's much more algae in these refuge habitats as a result of shy fish foraging in them in this treatment. Bonus little piggies there for you. Okay, so does fish behavior affect the spatial structure of food webs? Yes, shy fish produce the strongest differences in periphyton abundance between refuge and risky habitats. So these small changes to the behavioral composition can have pretty significant effects on the spatial structure of food webs, even in these tiny little enclosures. Okay, so just again to revisit the takeaways, I hope I showed you that juvenile coho salmon are more diverse than is typically appreciated, and that even subtle or small scale diversity can have pretty profound effects on population dynamics. Third, phenotypic variation interacts with key dimensions of landscape heterogeneity. We showed that stream gradient and position within the river network are very important. We think that low gradient streams nearest to the estuary seem to be most important in our system for supporting this non-natal rearing behavior. And finally, this is sort of an emergent idea that comes out of this, but managing for diversity in these strategies can improve conservation outcomes. finally, I showed you that behavioral variation can also affect food web dynamics. So these other things that we as ecologists think are important are also affected by trait variation that we see. Do I have time for my future directions? Okay, I'll try to rip through these really quickly. So a couple of directions that this has got me thinking. I want to try to integrate behavioral ecology and understand how it influences the life history diversity that we see in coho salmon. And this goes back to some work by Don Chapman back in 1965 for his dissertation work, which showed that aggression or territorial behavior in coho salmon was sort of one of these causes of fish moving downstream early. And then a follow-up piece in which he suggested that this territorial or aggressive behavior should change as a function of habitat conditions in the streams, where in faster flowing water, fish are having food constantly flow by them. So these juvenile coho salmon are just looking up and eating what they see. But in slow moving streams, fish have to kind of actively forage around the benthic habitat for getting fish. And that leads to this sort of aggressive territorial behavior where you get these strong social hierarchies that leads to aggression in fish. And some of the recent work that we've been doing has got me thinking that these things are at play in our system, and they haven't really been fully resolved since they were proposed back in 1965. So I just want to show you this photo of some fish that we captured recently in the sort of lower, slow-flowing portion of Willow Creek. So these are both young of the year coho salmon. So like roughly the same age, very different size. And what we found is that it seems like this was going on more often in these kind of slow flowing areas where you get these sort of divergent growth trajectories. And if you look closely at the tail of this small one, you can see what appear to be nip marks from aggressive interactions by other fish. So that's sort of how they display aggression, is by nipping the tails of others. So I'd like to kind of take a look at this just the old-fashioned way with a snorkel and a videotape, and just try to characterize how behavioral types change as habitat conditions change throughout the stream. And again, this sort of idea is consistent with some of our past results. So I showed you this figure here showing that fish that rear in the lower portion of Willow Creek have more variable growth trajectories than individuals that rear in sort of the higher portions of the creek where there's more drift feeding. And this is a new preliminary figure from a recent survey that we did, but in the same creek. So this is Willow Creek near the confluence, and then all the way up to the spawning habitat. And again, you can see that in the sort of what we're calling the natal habitat or the spawning habitat, you see much tighter dispersion of fish weight, whereas you see much more variable weight in the lower portions with much higher growth rates achieved. You can also see, so this is, sorry, they're not the same color, this is preliminary results, but along the same gradient, you can see this is effort standardized salmon mass within a single area. So you can see that the biomass of salmon that these areas supports is really different. So I kind of want to dig into this. Finally, one other idea from this paper, Don Chapman was like, yeah, we think aggression is really important, but also some of these fish just seem to have this sort of intrinsic desire to move downstream. And this has been sort of a theme emerging from behavioral ecology, where individuals have sort of these long-lasting quirks or personalities that seem to stick with them, that develop early in life and stick with them throughout their lives. So I'm really interested in doing some kind of coarse behavioral assays on fish in the hatchery that are to be pit tagged and released, and trying to understand if sort of these behavioral traits that we can measure pretty quickly in the lab correspond to their movement behavior in the wild. And finally, this is an important point. I realize we're almost out of time, but I'm really interested in understanding how we can manage for more diverse life history portfolios. So one really important aspect of that is going to be getting earlier life stage releases. So most of the data that I showed you today is on pit tagged individuals. And these are tags that are surgically implanted in fish. So you can't do them in these really small fish. And so our whole data set is missing juvenile fish. And another point is that the hatchery releases are typically done with larger fish. But there's a whole suite of life history portfolios that these tiny little fish can do in natural systems that are not being displayed in our system simply because the way that hatcheries typically work, which is raising fish until they're more stable at larger age classes. Juvenile fish are more vulnerable, they die more often, and so hatcheries typically like to raise them up to larger age classes before they release them. But there's some really neat tools for getting fish into the water earlier, and these include remote site incubators. This is a picture of one, but some of the folks that we work with at Goldridge and California State Parks are working to get these tools out into watersheds. And what they do is you stock them with eggs, and then you put them out into the tributaries where you want juvenile fish release. You pass water through them, and it incubates sort of in situ. And then you can release very small juvenile fish with very limited handling out into the water. So I'm interested in trying to understand sort of how these tools can be used to improve the suite of life history strategies that we can see in Willow Creek to sort of improve their dynamics. I'm working on trying to understand interactions between hatchery and wild salmon. Don't have too much time to talk about this now. And to try to improve our understanding of density dependence. Both of these are to maybe inform hatchery management. And then finally, there's a bunch of really cool restoration projects that are going on in the Russian River right now, including in Willow Creek and Green Valley Creek, which is another one that I mentioned today. And I would really like to be able to contribute to some of this monitoring to see if it's working, see how it affects some of these things that we think are really important for salmon, and hopefully contribute. A lot of these were well planned and have adaptive management components into them. So interacting with the folks that are actually doing the restoration projects to try to make them as good as they can be. So thank you all for your attention. Thanks to everyone who showed me around today. It's been a delight for me to be here. I'd like to just thank Mariska Obazinski, who's my colleague at Berkeley and at California Sea Grant, who spent decades building the monitoring program that I talked about today. My faculty advisors are awesome. You should meet them. Will, Greg, Andrew are all awesome people that I've worked with on the Russian River and are super knowledgeable about many of these things. Our colleagues at NOAA who have been amazing to work with, and then my lab. So thanks, and I'll take any questions if you have.
Very interesting. I feel like you talk forever, right? There's so much more. How's that time here? Yeah, if you have to leave, do so quietly. Of course, it's 4 o'clock. Don't have to stay, but hopefully you will for a little while. Ask a few questions here. I'm looking for a hand. I don't want to take Peter. I want to take no, Peter. You, please. I'm going to pass you the speaker though, because I hear you, right?
So what you showed us, the difference in nocturnal versus diurnal movements of fish in the steep streams and the shallower streams,
Yeah.
that's fascinating. So do you have any feeling for whether there are differences in visual predators, thinking of various birds, maybe some mammals in the two habitats?
Yeah, we've sort of been chewing on this a little bit. We're trying to write up these results right now. We're like, man, what do we make of these? Like, why are they moving into these high gradient streams more at nighttime? Like, it seems unlikely that, so the predators for juvenile coho salmon are piscivorous birds, and then things like pike minnow or piscivorous fishes. So it seems unlikely to me, just from my sort of ecological intuition, that those would be more abundant in these high gradient streams. And if anything, it seems like the mouth of Willow Creek would be a better place for them to hang out, or maybe not pike minnow, but yeah, it doesn't really seem like predator abundance is probably explaining that pattern. Again, I don't have any data to support that. That's just sort of my intuition. But it might have something to do with sort of the predator avoidance conditions within the stream. So like these low gradient streams get sort of sediment accumulation over time, and they get pretty turbid. And so maybe it's just they're less vulnerable to visually foraging predators because they're less visible to them in these low gradient streams, and so they can move in at any time of day. Maybe there is more structure to these streams that allow them to sort of hide from potential predators as they move in. I think that's possible. But again, it's just speculation here. Yeah. I hope that answers your question.
Questions? Yeah, maybe I'll ask a quick one as I'm walking. The non-natal rearing that doesn't work in drought years. Do you know why, or can you speculate?
Yeah, I can speculate. I think part of it might be that in drought years, the sort of very dynamic portion of Willow Creek, where there's like many different kind of complex channels, becomes really difficult for juvenile fish to navigate. And that's part of what some of the restoration projects are working to address right now. So they may just simply not find their way downstream. There might also be some sort of cue in the upper watershed, like it's low water years, stay where the water's cold. Yeah, we're just speculating, but those are some of the ideas we've had on it. Thanks, John.
Great, thank you. I really enjoyed that talk, and it really is fascinating how you've got really distinct populations, the natal and non-natal. And I was also really interested in what you said about how with the droughts, you don't really see that distinction. That got me wondering what you know about, like, to what extent these two behaviors are genetic, not just like to what extent there's heritability in it versus say something that, like you were saying at the end, that different organisms might, depending on the environment, have different behaviors or proclivities.
Yeah. No, that's a good question. Oh, so I'm supposed to repeat the question. So the question is this distinct bimodal pattern in natal and non-natal rearing. To what extent is that habitable? Is that, or excuse me, heritable? Is that the question? Okay. So we don't really have any information on that right now. Part of it. I think that's something that could be done with like whole genome sequencing, or just taking individuals. We know their movement trajectories. We have opportunities to clip their fins, and I think there is actually a bank of fin clips somewhere with potentially tied to pit tag detections, and so we could try to understand if through like a whole genome screen, genome-wide association study, if there's some relationship between that. In terms of heritability, yeah, other than that, we were slightly worried when we saw this, that this was just like an artifact of hatcheries where fish just kind of like take off downstream after the release from the hatcheries. But we do have detections on some wild fish that have been sort of opportunistically tagged over the years, and they seem to exhibit the same pattern. So at least there's no evidence that that's being selected against in the subsequent years after hatchery release. That's as thoroughly as I think I can answer that question. Yeah.
Thank you. There is a overarching question for fishes and vertebrates that sort of asks if you are a fast grower as a juvenile, like you're seeing in some of that diversity in Willow Creek, then you are potentially going to be successful and survive to reproduce. Not many people have data to be able to test that question, but it seems like you might be able to if you've got that huge variance in juvenile growth rates for when they're very small. And then you have a few, it looked like you had very few older individuals that were coming back, passing through the antenna. So can you say whether that very fastest growing portion of the distribution curve are your winners or not?
Yeah, that's a great question. So the question is, basically, is early life stage growth rate a good predictor of fitness or their ability to return as adults? And do we have data to sort of test that idea? And I'll just point back to, so this is that figure. So we only have 1 year of growth rate data right now. So this is like not very much growth rate data, and we only had very few adults come back. So there was not enough data to test that idea with what we had right now. But we had the same idea, and we are working on it. So the way that we're getting growth rate is through, because pit tide detections, right, they don't tell us how long the fish is. You have to get the fish in your hand and measure it. And the way that we do that is through smolt trapping. So as these individuals are on their way out to sea, you set up a trap, you capture them, you measure them, and then you send them away. Sonoma Water and California Sea Grant have been doing this for years, but the problem is that they had their smolt trap too far up in the watershed to capture these non-natal rears, and so that's why we don't have data on them. So we've been working with them to move their smolt trap down close to the confluence so that in the future, when they collect those data, it'll get sort of the full suite of life history traits within the river. And so we will be able to test that soon, and we have 1 year of data, and I think they're planning to do it. Hopefully I'll have an answer for you soon.
We maybe have time for one more question. I should have said to the people on Zoom that you should type your questions in, but you have to type really quickly now if we're still going to have time to answer it. But you can do it anyway. I can still give the question to Hank. Anybody else in the room here has a question? Okay, not going to wait too long. In terms of time, I think, Hank, you'll be here a little bit longer if we want to come and talk with you. Thank you so much. Really interesting talk. Appreciate you being here.
Thank you, guys.